Hello everyone, welcome back again to Eliza Noble Academy. Today's topic is modern concept of plasma membrane. Let me tell you that this particular topic is very useful for those students who are doing the MSc course in life science, either botany or you can see either zoology. MSc course in life science, either botany or you can see either zoology, because this particular topic is coming under cell biology, in which you will study the different types of cells, their physiology and different chemical compositions. So this topic is based on the modern concept of plasma membrane. Now here you can see this is the structures of a plasma membrane. Okay, this is called a fluid mosaic model of a plasma membrane. This is a fluid mosaic model because it is made up of different components of nutrients, like it is made up of protein, it is made up of lipids, and it is also made up of carbohydrates. So different nutrients, all together different nutrients are forming this fluid mosaic model of plasma membrane. Plasma membrane is a type of membranes which is found in a membranes of a living cells organism. Okay, so first let us see the chemical compositions. As I have said that it consists of lipid, fat, and all together protein. It is also proteins are also there, and carbohydrates is also there. So all together these nutrients, they combine and they form a fluid mosaic model. Now let us see this. The oligosaccharides. Okay, this is a carbohydrates component. It is of 1% to 5%. Okay, it may be attached to either the lipid or the protein. Here, like carbohydrate, protein. This carbohydrate, as it is said, the oligosaccharide, is it either connected to the lipid or to the protein? Okay, there is also a wide variations in the lipid protein ratio between different cells membrane, that we will see in the next slide. Now we have come to know the chemical compositions of a plasma membrane, that it consists of, or it is basically composed up of lipids. Okay, these are lipids. Protein, these are the different proteins. You can see the cursor which is moving. These are the different proteins, and these are the carbohydrates. These are the carbohydrates: glycoprotein, glucose, glycolipid. Why it is glycoprotein? Because carbohydrates and protein. And why it is glycolipid? Because carbohydrate is attached with the lipid. You can see this is the lipid, and here this carbohydrate is attached with the lipid. That's why it is called as glycolipid. And here the carbohydrate is attached with the protein. This protein, focus onto the cursor. This is the carbohydrate and it is attached to this protein. That is why it is called as glycoprotein. As we have seen in the compositions, that this plasma membrane is composed up of fat, that is lipid, protein or carbohydrates, and carbohydrates is either attached to protein or it is attached to a lipids. That's why if it is attached to lipid, it is glycoprotein, and if it is attached to protein, it is glycoprotein, and if it is attached to lipid, it is glycolipid. Now let us see the different composition. See, this is the lipid fraction of plasma membrane. Okay, the main constituent are phospholipids, cholesterol, and galactolipids. Their proportions varies in different cell membranes. This particular image is the enlargement of this image. This one. Okay, these are phospholipids. This is a polar head and this is a polar tail. Polar head is hydrophilic. because it is submerged in water. That is why it is hydrophilic. Hydrophilic means it is submerged in water. And this is the polar tail, which are hydrophobics. So these polar tails are particularly the fatty acids. Okay, the polar tail, this tail. These two you can see in blue. These two are the polar head, and these polar tail are the fatty acids, which are the hydrophobic. Hydrophobics because they are not submerged in water. Let us clear in this slide. Lipid molecule is basically consists of two parts of heads. Don't get confused. This image is the only, okay, this image is the only enlargement of this phospholipids. Polar head with polar tail. So it is consist of head. These head are glycerol. Yes, as I have said that this hydrophilic polar head are glycerol, and this tail are the polar tail, which are the fatty acid. These two polar heads are the glycerol, and this polar tail are the fatty acid. So basically this is the structures of a plasma membrane. Okay, and the most important thing in this is that first you need to know that what is the compositions of this plasma membrane. So we all know that it is made up of lipid, protein and carbs. Carbohydrate is either connected with the protein, that is why it is glycoprotein, and if it is either connected with the lipid, it is called as glycolipid. Okay, this is the main compositions. And this phospholipids, this is a polar head, which is hydrophilic, and this is polar tail, which is of fatty acid. Polar head is of glycerol, and this polar tail are of fatty acids. Okay, main. Now let us see the protein structures. Okay, so this is the mosaic fruit model of a plasma membrane. See, phospholipids, polar head, and this is the polar tail. In between this phospholipids, proteins are embedded. Okay, the proteins are embedded in this image. You can see that the protein are here, and this is the brown color protein. This protein, these are either intrinsic or either extrinsic. In this slide you will be very clear what is intrinsic and what is extrinsic. Okay, so here this is an intrinsic protein. This protein is located in the integral part of this plasma membrane. That is why it is called as intrinsics. It is located in the integral. It means inside integral. And here the extrinsics are at the peripheral, here or here. Oops, sorry, I just shake this line. Here you can see here or here. So those proteins which are at the peripheral, means at the edge of the plasma membrane, they are called as extrinsic protein, and those protein which are embedded in between the phospholipids, they are called intrinsic protein. This is how the protein is being structured in the plasma membrane, and it is located. See, we have seen the structures of protein. Now let us look into the effect of osmosis. Plasma membrane, we know that is it a kind of membrane which is present in all the cells, organs of a living cells. So what happened? How the effect of osmosis is happening in the plasma membrane? First of all, what is osmosis? Osmosis is the spontaneous movement of molecules from its higher concentrations to its lower concentrations. Now, what do? We drink. Yes, we drink water. Okay, we drink water. Let me tell you, when we drink water, how do the water hydrate our organs? How do it hydrate our organs? Because at the outer layer of our organs, it is the membranes are there present, cell membranes are there, organ membranes, that is plasma membrane. How do the water get inside our body? It is through the process of osmosis, plasma membrane. Now, how do the food particles? Now, what happened? See this the effect of osmosis. It consists of 3 different types of solutions: hypertonic solution, isotonic solutions, and hypotonic solutions. Now, what happened in hypotonic solution? The cells inside the cytoplasms, they get shrink when the solution is hypertonic. It means the cells get shrinks when it's the fluid inside that particular cells is moving towards the lower concentrations. That is how it is shrink. For example, if we are taking the example of a raisin, we soak the raisins for overnight, and during the morning hour it get swallowed. Why? Because the water contents goes inside the raisins, and which it is moving from its higher concentrations to its lower concentrations. We know that water is a universal solvent. Now it has entered into the raisins, then it swells up the raisins. Similarly, the same process is taking place when hypertonic solution is there. This particular cells gets shrink because the water contents comes out from the plasma membrane of this particular cell. Now, what happened when that particular cells need water? Isotonic solutions. Here what happened? The concentration is equal at the outer environment of the cells. The amount of the molecules is equal to that amount of the molecules which is present inside the cell. That is why it is neither shrink or it is neither swallow. This is called as isotonic solution. Hypotonic means during the hypotonic solution, the surrounding molecules are moving from the higher concentrations to the lower concentrations. The higher concentrations environment is created near the surrounding of the cell organs, and the lower concentrations environment is inside the cell organs. So what the molecules will do, the molecules will move from the higher concentrations to inside the cell's organs. So this solutions is called hypotonic. We have seen the 3 different effects of osmosis during the plasma membrane transporting process. Osmosis is nothing but the movement of it is the movement of molecules from higher concentrations to lower concentrations. Hypertonics, what happened inside the cell organelle? The environment was here inside the cell organs, the concentrations was high. So from here the molecules has diffused to the lower concentrations. Isotonic, here the surrounding the molecules and inside the cell organs the molecules were equally distributed. That is why it was balanced. In this hypotonic case, the surrounding molecules were moving from the higher concentrations to the lower concentrations inside the cell organ. Now we will see that how diffusion is facilitated through different proteins. So this is the plasma membrane. This is the plasma membrane. You can see these are the phospholipids, glycerol and fatty acids. Now how osmosis and diffusion takes place. In the previous slide we have seen the osmosis, how molecules enter the cell organ, and how they are entering the cell organ, and how they are extracted out from the cell organ. This basically is happen like this, and helped by this carrier protein, which are the facilitated of diffusions or active transported of molecules from the outer environment to the inner environment of a cell organ. What is happening actually in this process? The carrier helps the molecules of metabolites along the directions in which that molecules ordinarily move. So basically, those carrier are the protein. They are the protein which will selectively. Why it is called as permeable plasma membrane? Permeable plasma membrane. It means because it is not like that any molecules can enter the cell organ. No. This protein will be helping the molecules. And what type of molecules? That too selected molecules. Like, let me tell you an example, that we have a gate in our house or in a compound. Why that gate is there? So unknown and strangers cannot enter our house. That is why that gate. And in that gate you will have the guards. That guard will be allowing the person. Accordingly, he is instructed that this person should be allowed to come inside. So imagine that this plasma membrane, okay, this is the barrier, or you can say the campus, or you can say the railing, okay, and this side is your home inside, and this is the outside. So consider this as the railing of your home, and these carrier protein considered as the gate. Okay, consider these as a gate and the carrier protein as a guard. Now these guard, what they will do, they will only selectively allow the molecules or that persons to enter your home, like that. This is process because it is permeable. It will not allow all types of molecules to enter inside the cell organ. No, only selectively cells will be allowed to transport it inside the cell organ. So basically, the carrier protein are helping in transporting the molecules, and that too selectively molecules. So this was the general outline or a modern concept of plasma membrane. Now there are different structures of plasma membrane based on different scientists, like the lamellar model, which is a biomolecular leaflet model of Danieli and Dawson, and the unit membrane hypothesis J. David Robinson. And this concept which we are dealing with is the fluid mosaic model. This one is the fluid mosaic model of plasma membrane, and the lamellar model of plasma membrane is somehow looking like this, which is proposed by Danieli and Dawson. And fluid mosaic model is this, because it is composed of different nutrients, protein, carbohydrates and lipids. It is look like fluids. That is why it is called as fluid mosaic model. That was proposed by Singer and Nichols. So this entire slide was a slide of the modern concept of plasma membrane. I hope this video would be helpful to you all. Thank you. Please do like, subscribe and share the video.